Welcome everyone to a think tank that is centered around some new or common life detection techniques. So the way we run this is a little bit up to the committee. So what we decided is to do something a little bit different, and we decided to do one longer talk and then a little bit shorter talk that each will be followed by questions that are centered around Raman spectroscopy techniques for life detection. So first speaker will be Dr. Scott Perl, who is an astrobiologist and geobiologist at JPL since 2012. He is the co-PI of the JPL Origins and Habitability Lab, and also was the investigation scientist for the CRISM instrument on board the Mars Reconnaissance Orbiter. He has also previously worked on the Mars Exploration Rover, Mars Science Laboratory, and now Mars Perseverance, the 2020 mission. And he has been involved with several other life detection science concepts over his career. He is also a research affiliate at the LA National History Museum and is a visiting professor at Caltech. And he specializes in vibrational spectroscopy techniques for microbial life detection under in situ conditions as well as laboratory conditions. And his particular interest is the habitability in life detection in high salt environments, I think, if I can sum it up, including Mars and Europa analog environments. So take it away, Scott.
Sounds great. Thank you, Roland. I'll share my screen to kick it off. Okay. So assuming folks can see, we'll just start off. So thank you for the introduction. Thank you for having me for this meeting. And I'm happy to discuss this in terms of both this talk as well as our think tank section later on. But to kind of kick it off, I wanted to make sure that we're on the same page regarding how we categorize life. And so something that both the NASA, in terms of exobiology programs and how we go about life detection, we're still in the habitability realm a lot of the times when it comes down to us trying to look at features that could have supported life. And so as we go from the habitability regime to actual life detection, when it comes down to biology on other worlds and what this means regarding a separate evolution, we have to be sensitive in terms of what we can measure with regard to our instruments, but what is a bias for our own terrestrial kinds of environments. And so when it comes down to being able to understand how we actually look at samples. So by design, Raman cannot do any microbiology in terms of that kind of data set. We're not saying that Raman can make cellular community actual identifications. It's more about what the components are of actual biological systems. And so a lot of the work that I do involves microbial pigments. And so the pigments themselves show a kind of adaptation in terms of photobiology to high UV environments. And so on Earth, this is UVA and UVB. On Mars, potentially, this would be UVC. And so the idea of being able to use Raman to know the vibrational sensitivity of these pigments, we both get an idea of a cell adaptation from these communities, but also how the actual pigments themselves are actually preserved inside the rock record, as well as the mineral record. And so it's something to the effect of when we're able to actually use Raman to measure organics, that doesn't necessarily mean we have extinct life or actual extant life. But if we had biology in some preserved system, surely there would be organics. And so being able to denote what that relationship is very important in terms of Raman spectroscopy. And so kind of zooming in on this figure on the right, this is from our paper from 2021. What this is showing on the left two green columns, this is showing both signs of life with regard to what's preserved inside the mineral record and actual life itself. And so it's something that we can use Raman on these samples to get out a Raman spectra of features we're looking at, but the actual identification of what's preserved inside this record in terms of the salt mineral can't be done with Raman. So having Raman alongside some kind of microbial technique to make some kind of identification, that's the ability where you really strengthen the argument to understand this is who is living there, this is what the organics are, this is how they're preserved, and these are the cells that are in these environments. And that's more for the extant life. If you're in the extinct life category, more so on the right side of this pendulum diagram, you've lost all evidence of cells. And so pigments in this case is something that can last over geologic time. We've seen pigments as old as the Permian age, if not older. And so on the right side of the column, we've lost all the biology, but what could be from life actually remains. And so the idea of having fluids preserved in terms of some kind of paleoclimate, this would be able to tell us, in terms of us assuming that there's no contamination, what's actually preserved inside the mineral record. And on the very right side of this is just showing a mineral in this case. And so all four of these columns, we have the same sample, but the way that these are being measured will give us different results. And that's important in terms of Raman techniques. So I mentioned before in terms of pigments, and I tend to look at high salt environments, very, very high salinity features in terms of brines and minerals. And so what I'm showing here is from a paper that's in revision right now. You're looking at the Great Salt Lake in figures A and B, and these are areas where you have some kind of pigment preservation in, these are both in the modern time frame. You can see there's a small little pigmented section underneath some salt crust here. This is sodium chloride that's found right near the Great Salt Lake. And for folks that have been keeping up, the Great Salt Lake is actually shrinking. And if things don't change in terms of the next few years, we'll actually probably lose the lake in the next few decades, maybe even sooner. On the right side, figure B is calcium sulfate that is formed from groundwater upwelling permeating through some of the actual regolith also near the lake. And so the groundwater source here is the actual Great Salt Lake brines. But you can see the different chemistries as you're going forward, where you're going through sodium chloride to calcium sulfate. Both have a pigment structure, both are Raman sensitive, but you're looking at biology here with regard to their response to the Great Salt Lake photobiology. Whereas out in terms of Death Valley, you have brines that have a very, very high solar flux. And so not only do you have, in terms of more time of the day, you have sunlight hitting both the brine pools and the salts, but you have features that throughout the year have a lot higher sensitivity or a lot higher exposure to both UVA and UVB. And so you have the same lithology for both of these sites. Raman can tell you a lot between these two, as well as the preservation, but getting an idea of who is making these pigments and what they're actually doing in terms of these communities, Raman can help aid in that, not actually identify what's there. So when we bring this to Mars as well as Europa, we have a certain set of questions with regard to extinct and extant life. And so for the Martian example, we're looking at, and this is just on the Martian surface, we're looking at features that have been preserved over the last 3.5 billion years. And so what is the threshold in this case of that preservation? What would we actually expect over geologic time in terms of what's been preserved? If it was preserved, from the time of formation to what we're looking at now, if that preservation is immaculate, what do we actually expect over geologic time? And so this becomes three sub-questions of you're at the mercy of your source biology. Whatever is preserved at time zero is what is the maximum that you'd expect over geologic time. And if you have poor preservation or if your features are exposed to the Martian atmosphere, especially modern Mars, you might lose a lot of that preservation. And so these three questions dictate a lot of the research paradigms that are in my lab. And this is, again, for extinct life and on the Martian surface. For signs of life that are more extant and actually present regarding a solvent, you have a similar set of questions, but more so in terms of radiation and ice. And so with regard to psychrophiles and halophiles in this case, your threshold involves extant communities. So the communities that are living in these systems. And obviously, if you're in a frozen or highly salt system, your biodiversity might be very, very low, but your volume of biomass might be high. And that's something to take into account with regard to actual community adaptation in these planetary features, if life was ever present. Your source biology now has to have some kind of genes that are expressed to not only live in the salt, but live in the radiation and live in the ice. And what would that look like right now? And so this mindset of actual microbial adaptation brings our threshold of actual habitability a little bit higher into areas where we wouldn't expect life to live. But we have to take into account if you're talking about extant life and you're in a system that is highly volatile from our own kind of biosphere, life would have had to contend with that for a very long period of time, such that it might have been able to adapt to those environments. So the paradigm might be a little bit easier than what we give it credit for. So with regard to extinct versus extant life, this is also from our paper from a couple years ago. When we're looking at the ocean worlds, we're essentially on the left side this diagram where all of our extant life, you have features that would actually respond to our experiments. So if you have microbial communities that are alive and well, there would be some stimulus response, whether it be actual light or chemistry. All the Mars missions up until now are on the right side of this diagram, where signs of life are all that's left potentially, but there's no biology remaining on the Martian surface. However, in terms of the Martian subsurface, there are features where if we were to get to areas where you have a closed aquifer, something that has not had access to the Martian surface, you actually, and this is based on the fact of us being able to go and actually validate that there was authigenic water on Mars in terms of the Martian subsurface, however deep that would be. You can treat a Martian aquifer almost with the same rules as we do for the ocean worlds, right? But you have the time frame here to understand that you have a rocky planet, you have substrates, that you had water rock interactions, and so the probability of habitability and life does actually increase. So it's something to keep in mind versus Mars is the ancient dead life, and all the ocean worlds have the active life. There are features that do cross between the two. And so the take-home point here again is we're still trapped on the initial biological volumes, and this is more of a kind of Mars sample return point of view rather than actual extant life in the Martian subsurface. So we've actually done this where we're looking at old signs of life inside salt and what's left over. Charles Capel and I published a paper in 2020 along with other co-authors here that went into lipid actual preservation within 253 million year old salts. And so that's on the tail end of our geologic time, whereas in the modern end, in terms of the Great Salt Lake and other areas in Death Valley, what is our initial time zero of preservation as cells are being preserved? And so there's a giant gap in the middle in terms of preservation, but also with regard to yield of what remains, but also with regard to what we can actually validate to actually say that it was these species that were living here rather than these are the organics that are left over. It might have been from life, it might not have been. And so being able to pull that paradigm more to the geologic time to get a much higher fidelity amount of data is extremely important. So what I'm showing here, just really quickly, are set up abiotic experiments that we did quite a while ago to map what the biology does with, or at least what it would do if it was actually here with regard to pigments. And so what I'm showing here are 3 brine droplets. 7A is the one I'm going to focus on. And so the figure on the top right is just an image of 3 or 4 salt crystals. You can see the cubes on the right, and then map the Raman spectra of those certain carotenoids to the Raman spectra, and then made the maps as we're actually forming salt. So the end part of the process, where you can kind of see it's actually evaporating counterclockwise, the salt crystals that are here on the left, there's one of those pockets that we actually measured. And so what you're seeing here is, if it is green in terms of this PCA, all 3 to 7 carotenoid peaks were found in that salt crystal. So spatially mapping the carotenoids to the salt crystal, anything that was red is a carotenoid that was found outside the salt crystal. Now, this is a highly manual process, and it's something that we do trust as we're doing this manually, but it is time intensive. So being able to do this on a much faster scale for our brines is in work, but it's something that we want to still make sure that we have the process well done. And so what you're seeing here is the cumulative spectra of what's showing here to make up that PCA. The point is that here, the time it takes to go from your droplet to the mineral is your preservation metric, right? If it happens quickly, you can assume that you've ported over a lot of those communities together. When you have high salt, in this case, you can actually break carotenoid bonds. And so while trying to replicate the Dead Sea, we did this for the abiogenic experiments where we broke some of the beta-carotene bonds here. And when you fracture those carotenoid bonds, you still have a Raman signature, but now they're vibrating all over the place. And so I note this akin to if you had a tin jar of marbles and there was 100 marbles in there versus if you had a tin jar of 4 marbles and you shook that around, that would sound very, very different. You can tell there's a lot more, but those vibrational modes are going to be all over the place and few and far between. And so you can see even here for just 3 carotenoid peaks, you have a much higher intensity, about 45,000 counts. Whereas when you break all of them, you get down to about 700 counts. And so mathematically, you can resolve this. And this is something that we did. This is a paper that's in review right now. And so you can mathematically calculate what the broken bonds are. It's very difficult, but you have to also know what the original bonds are. So it's something that you both get a paleoclimate constraint. So I know that at 46 or 44% salinity, this happens to beta carotene. So not only do I get a kind of salinity max for what the Raman signature is showing me, if I was to ever see these broken bonds, or at least these kind of moderate to major peaks, I would know that we're talking about a broken set of carotenoids that are at these salinities. So a very specific example, but still very useful. I won't really spend time on this in terms of actual Martian habitability, but we are looking at all these salt crystals and all these actual former brine features, because on Mars, these are all the hotspots where we would land with a future mission. So every mineral you see here, in terms of both the actual salt mineral diversity, as well as the fact that you have to go through some actual kinetics to get to what we're seeing here. These features all touched ancient surface waters, and so whether it's a primary mineral formation or something secondary or tertiary, everything you're seeing here in terms of these icons has touched some actual ancient Martian water. How much water it was in terms of volume, the chemistry of those brines, and then actual duration is obviously something we can back calculate in terms of these. And this is also already 2014, so there are updated CRISA maps. This is just the most recent one that folks are familiar with. I'll kind of skip through this quickly, but to make sure that we're on the same page regarding physical biosignatures and chemical biomarkers. The physical biosignature is something you're able to see in terms of the naked eye. A chemical biomarker is something that you're able to measure. And so carotenoids, fortunately, fit the bill for both. And so the paper that we have in revision now looks at what is being ported over from the Great Salt Lake water column to the salt crystals nearby. And so what we're seeing here, essentially, using my hand for scale, we see pigments that are preserved inside salt crystals, and then we're able to make the measurements using Raman on what's preserved inside those fluid inclusions. And so how much of the lake, of what's living inside the lake, is actually being preserved inside the crystals themselves. And so having the actual ability to know not only who is living there, but what they're producing in terms of Raman. And I'll plug this. This is our book chapter that we had in terms of the first Great Salt Lake biology book and the first astrobiology chapter of that. So this is something that went out in 2020, I believe. And so the chapters, I think, are available online. Folks can check it out. But what I'm showing here is that you have a relationship between the mineral lithology and who is preserved inside those components. And so just in terms of the calcium sulfate example, there is a ton of iron inside the clays that are found inside calcium sulfate. We have microbial communities, specifically these one or two halophile communities, that are only found inside portions of the mineral that do have iron. So we can make the assumption using Raman and the actual 16S sequencing here that there is a relationship between the clay beds that have iron and the microbial communities that are only found inside parts of the mineral that do contain iron. And so it's a good one-to-one ratio to understand what it looks like spatially with Raman and what it looks like in terms of microbial communities that are there. When we're talking about preservation of extant life, kind of hammering the point home is when you do preserve a microbial community, excuse me, you also preserve the microenvironment of where they're found in. And so I show here cells that have a high cell motility. It's about 10 to the 5, 10 to the 6 cells, but you're in a microenvironment that is a version of what was preserved from the lake. And so in this case, this was from our original campaign several years ago. And so having Raman spectra of cells that are actually modal and present here not only gives us the idea of how they're preserved, but the volume that's there. So you get the spatial awareness of what's preserved, as well as the Raman spectra of what that chemistry looks like. I'll go into very briefly, because I'm kind of running out of time, with regard to the array of brine and salt mineral work here. We're looking at single cell detections. We're looking at Permian brine preservation, as well as isotopic measurements of low biomass systems, how these carotenoids can be irradiated but still preserved in terms of the Martian environment. We have a subgroup in terms of a Center for Life detection effort that was a few years ago, where crystal habits as a whole is a good sign of where you'd find these biosignatures, and then constraints regarding subsurface brine systems. And so I will kind of go through and want to thank all of my colleagues over the last few years / proposals and all of my students in terms of the Origins and Habitability Lab. And I will move over to the next speaker.
Thank you, Scott. And now we have Claire, our own very own from our steering committee. And I believe, Claire, you're going to also talk about your experiences with Raman and live detection measurements.
Yes, I am. Oh, sorry.
You go ahead and flash your slides, and I'm gonna quickly introduce you.
Yeah, thank you. I'm trying to
for the YouTube channel.
Yeah, sorry, I'm trying to get my computer to function here. Okay.
Okay, so Claire Marshall has a PhD in chemistry at the University of Technology in Sydney, and after her PhD, she went on for a postdoc at the Australian Centre for Astrobiology at Macquarie University in Sydney. After that, she obtained a Queen Elizabeth Fellowship in the School of Chemistry at the University of Sydney, and she's now first has been an assistant professor and now is an associate professor at the University of Kansas for 16 years now. Take it away, Claire.
Yeah, thank you. Thank you so much. Thanks for inviting me to give a talk here. And so I'm talking today about doing a feasibility study for what we call Raman optical activity, and I'll explain what that is in the next few slides. So this is not necessarily an instrument building study, but looking at a feasibility study utilizing Raman optical activity to see, firstly, can we detect and then discriminate amino acids and use this information to feed into instrument design. First of all, sorry, you have to excuse my voice. I have what is called the KU cough that is going around. So pardon me if I start bursting out coughing. I'll try and do my best not to. So first of all, I'd like to introduce my old master's student who's now moved on to Clemson University, Will, and Will did all the lab at a company called BioTools, which I'll talk about that in a moment. So getting my computer, okay, so there we go. Okay, so this audience is very familiar with amino acids, and the big question is, are amino acids biosignatures, and should we detect the presence on other planets outside of Earth? So as we all know, amino acids can be formed both abiotically and biotically. And this nice diagram from Creamer et al. in 2017 demonstrates this quite nicely, where here we have Escherichia coli and the presence of amino acids that can be found that are biosynthesized by that microbe. And as we all know in this audience, that we can also find the presence of amino acids, for example, in meteorites. So therefore, in these cases, it's not merely the presence, but rather the chirality of these molecules that is indicative or could be indicative of a biological origin. So as we know, we could use amino acids as a biosignature based on their chirality. So we have left-handed and right-handed amino acids. And here on Earth, predominantly, life utilizes the left-handed amino acid as a building block to make proteins. And there are some exceptions, as there always is, but predominantly, we tend to utilize left-handed amino acids as a biosignature. And if we look at the current Raman flight-ready systems, or systems that are actually roving around on Mars, and systems that presumably, I can't remember when the next date for ExoMars is, but presumably in the next few years, the Raman laser system will also be roving around on Mars. So if we look at the Raman subsystem in Sherlock, the Raman subsystem in SuperCam, and RLS, they don't have the capability to distinguish between the D and L enantiomers of amino acids. And Raman optical activity spectroscopy is a relatively new method and it combines the variability of scattering experiments with the structural sensitivity of chiral spectroscopy. And interestingly, today, ROA spectroscopy has only been utilized in the fields of pharmaceutical chemistry, biochemistry, and biophysics, but has yet to be applied to astrobiology. And so what is Raman optical activity? How is it different than Raman spectroscopy? And because I only have a 10-minute talk, as much as I love talking about math equations, I'm just going to put some relevant equations here. And some of these explain what ROA is, but also some of these are actually needed to be considered for instrument development and design. So first of all, Raman optical activity utilizes circularly polarized light. And that light is scattered when we use unpolarized light, and we have an electro-optic modulator that then scatters circularly polarized light. We impinge that upon the sample, and the sample either scatters right-handed or left-handed scattered light, or circularly polarized light. And this can be observed experimentally as the circular intensity difference, and that's defined by this equation here. So this is the incident scattered polarized, circularly polarized light, and we have the right scattered circularly polarized light minus the left scattered circularly polarized light. And here on the bottom, this is Stokes or conventional Raman scattering, where we have the summation of right and left circularly polarized scattered light. So the ROA component is looking at this numerator here. Now, unlike conventional Raman spectroscopy or Stokes Raman scattering, the light interacts with the electric dipole or polarizability tensor. So this is a polarizability tensor. It's a second-rank tensor. Some people refer to it as the Raman tensor, or some people refer to it as the polarizability tensor. So that's what you expect in terms of a substance being Raman active or Raman allowed or not allowed in conventional Stokes Raman scattering. However, ROA is a little bit more painful in terms of instrument development, because ROA, in addition to the polarizability tensor, is also sensitive to contributions not only from the polarizability tensor, but also from the electric dipole, magnetic dipole, optical activity tensor, G prime here, and the electric dipole, electric quadrupole tensor, A. And so I'm not going to go through this equation, but needless to say, in addition to the polarizability tensor, having these other tensors really weakens the Raman effect. So we all know the law of Rayleigh scattering, which is 1 over lambda to the power of 4, and that holds true for Raman scattering. When you're talking about ROA scattering, it is not 1 over lambda 4, it's another order of magnitude weaker. And so that's why it's important to introduce this equation in terms of looking at these G and A tensors, because it makes it a much weaker Raman scattering phenomena. And here's just a little optical train or pathway diagram. So there's only one company in the world that manufactures a ROA spectrometer, and that company is BioTools, which funnily enough is located in Jupiter, Florida. And this Picasso that I've been awarded is looking at the development of ROA for Ocean World mission. So it's kind of funny that the company is located in Jupiter. And this is the current ROA, and it's the only commercial ROA spectrometer available. And it's called the Chiral Raman 2XTM. And here's a little optical train. And I'm not going to go through it in great detail because I'm probably hitting the limits of my talk time, but a few things I will point out that are salient for instrument development. So first of all, you could see that we have a polarizer here, and we have this electro-optic modulator, and we have a bunch of apertures here. And you'll notice that the sample cell is located here, and we do collect in 180 degrees backscattering. That's the most efficient way for ROA spectroscopy. And you'll notice that we put in a polarizer and this electric optic modulator. So we're getting a huge incident laser light signal drop in intensity because we have these two optics here. And so basically this current system is built on a unpolarized laser, and then it goes through a polarizer, and then you circularly polarize left and right-handed scattered light in this electric optical modulator. And you obviously get a lot of signal drop, and then the way that the geometry is here, it's not that conducive to maximize the backscattered Raman light. And again, you go pretty much through normal optics as you would for conventional Raman spectroscopy. So it's just worthwhile pointing out the front end of the optical train to actually induce this left and right-handed scattered light. Okay, so the objectives for today, I'm not going to go through all the work that we've done, but I'm just going to go through looking at, firstly, establishing if ROA can actually discriminate between D and L enantiomers of amino acids. You may be surprised by this. I was when I first started this project. There is absolutely no literature that actually shows ROA can do this. So presumably, yes, it can, and I'll show you that it can, but it's interesting that there's this gap in the literature. And then the most important thing, particularly for instrument development, is to establish if our ROA measurements can be performed on likely amino acid concentrations that you would experience in ocean wells. So this is what we call the limit of detection. And so this is an important part of the study. Using the current benchtop configuration to see what the LOD is for that, and then using that information to feed forward into instrument design in terms of revamping the whole optical train. So here we go. Here's the big answer to the question. Can ROA distinguish between L and D alanine, for example? And here on the top, we can see conventional Raman spectroscopy. And this instrument manufactured by BioTools can collect conventional Raman spectra, and then it can also collect ROA spectra. And so anyway, here we have a mixture of L and D alanine. And if you look at a conventional Stokes-Raman scattering spectra of just L or just D aniline, the spectra, you could overlay them, they look exactly the same. But here, if you have a mixture of both, you collect a Raman spectrum, and yeah, sure, it's aniline, but is it D or L? And if you look at the bottom spectra below here, this is the ROA spectra for a mixture of, as you can see here in red, D aniline and L aniline. And as you can see, this is bisignate spectra, and the different enantiomers either have a positive or a negative signal for that particular vibrational mode. So here, if you look at in red, you can see negative, positive, positive, negative, positive, and so on and so forth. And this corresponds to D aniline. And again, if we look at the spectrum in black, we can notice that we have positive, negative, 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 and positive, and so on and so forth. And this corresponds to the spectrum for L aniline. Now, a few things to notice here. You'll notice that this Raman spectrum looks pretty good. Nice signal to noise and nice intense vibrational features. If you look at the ROA spectrum below, this took 4 hours longer to collect yeah, 4 hours longer to collect than the top spectrum. And that's just simply due to a function of a weaker Raman scattering phenomena. And you'll notice that this is quite noisy. And also in this higher energy region here, we lose a lot of feature. We don't actually resolve that from the baseline. And as we move towards lower energy, again, we're not resolving as much feature from conventional Raman spectroscopy with respect to ROA. But nonetheless, that said, we can actually distinguish between D and L alanine. So what happens when we progressively serial dilute this solution to try and achieve perhaps somewhat getting close, or maybe as close as we can, to realistic concentrations? So again, the top spectrum here is conventional Raman spectroscopy. And we notice that we start out at 1.8 molar, and we progressively serial dilute down to about 0.5 molar. And clearly, obviously, we could see that we get a reduction in signal with decreasing concentration. And here at 0.5 molar with conventional Raman spectroscopy, we still have a reasonably good limit of detection. We could still see nice resolved weak to moderately and strong intense Raman bands. And again, if we compare with what we can do with current ROA technology, if we look at 0.5 molar, we're kind of not in that much good shape with respect to limit of detection. There are some features that you can see at 0.5 molar, but also we're missing a lot of other features. And here's just looking at taking the natural log of the signal to noise and the natural log of concentration to come up with a LOD quantification graph. And this just lets us know the limit of detection. And what's actually more important is the limit of quantification. And so the limit of detection is just detecting, can we see little Raman bands or not? And the limit of quantification is, well, yeah, we can see Raman bands, but can we actually assign them to a particular vibrational mode that we can assign that particular amino acid to? So in terms of the current optical train or the current configuration for ROA technology, the limit of quantification is about 0.028 molar, which is not realistically in the nanomolar concentration that we might likely expect to see for amino acids on ocean worlds, or Lake Vostok down in the Antarctica. So takeaway points from the work done so far: the current design of the chiral Raman 2x-TM ROA spectrometer is not conducive to detect ocean world concentrations of amino acids. So that's valuable information to ascertain, and knowing this means that for the next part of this work, there's going to have to be a new optical train design, and also sample presentation and a SIRS probe would be required as well to get that surface enhanced Raman scattering, given that we're an order of magnitude weaker than conventional Raman scattering. So the idea would be to redesign the optical train and design a SIRS probe to try and boost that signal to compensate for that order of magnitude weaker Raman scattering. So with that, I'd like to thank the NASA Picasso grant that's funded this work, or still funding this work. And I'd also like to thank BioTools going down there and letting us use their instrument. So thank you. Oh, should I stop sharing the slide or?